Phenome UK is a UKRI funded technology touching life network. This is a program that spans the research councils. Ours is managed primarily by BBSRC, and the whole point of the Technology Touching Life program is that it aims to bring people together from the engineering and physical sciences with people from the life sciences to try to generate new technologies and methods that will allow people in the life sciences to address core scientific issues. So our network is focusing on plant phenotyping. The argument being that if we can provide the UK with good plant phenotyping facilities and infrastructure, that will enable people to make big inroads into problems around food, crops, climate change, and so on. So this slide is just showing the text from the call so that you can read it and see what we're about. It's about technologies for measuring quantitative structure and function of plants. We have two broad aims. One is to help UK scientists gain access to the technological methods and tools that they need. So we're about developing new techniques, evaluating new techniques, and so on. And we have a second goal, which is to find out what the UK needs. So there are a number of plant phenotyping initiatives underway internationally, and in particular in Europe. There is a thing you may have heard of called Emphasis, which is an S3 project aimed at creating a pan-European plant phenotyping infrastructure. So we're focusing very much on what the UK needs, hopefully to make it better, but also to prepare us for the coming of Emphasis. So we have a number of instruments available to us in Phenome UK, and the classic networking tools of an annual conference and a variety of different workshops. We identify outreach workshops as those that help us specifically go to other communities and try to bring them into the area. And we have the idea of issue-based workshops like today's, which focus on particular questions relevant to the community. Something that's perhaps unusual for many networking projects is we also have the ability to fund pilot projects. So we have a fairly light touch review process, and we can fund projects up to about £25,000. Okay? So that allows us to actually get some work done. So far, we've funded 8 of these projects. There will be a third call for the last set early in the new year. Okay? So you can see broadly how we intend to work, from people meeting in the larger meetings to some discussion in the smaller meetings to actually doing some projects. Okay? Our principle behind setting up the network is if you want people to collaborate properly, then they have to do a real project, and that's the best way to do it. So if you would be interested in finding out more about us, our website is there. We have an email address which will go to Claire, who's behind the scenes. And if you would like to get involved in anything, have a look at the website and get in touch. Okay. So with that, I will stop sharing my screen and I'll hand over to Harry, who's going to introduce CHAP and share the rest of the morning. Thank you.
Hi there, thanks very much, Tony, for that introduction to Phenome UK. I'll just quickly start sharing my my screen. Works. Is that working for everyone?
Yeah.
Great, thanks. So yeah, welcome to this this morning's workshop, co-hosted between CHAP and Phenome UK. So I just wanted to give a brief overview of who we are at CHAP first. So we're one of the 4 agri-tech centres that were funded a few years ago now, with a £90 million investment from Innovate UK. And so we sit alongside our sister centres, CL, AgriEpi and Agrimetrics, who work in different areas of the agri-tech arena. And ourselves at CHAP, or Crop Health and Protection Limited, we're a not-for-profit government-funded organisation that are focused on crop and soil health and protection, and fundamentally how we can manage threats to crops with fewer interventions and lower environmental impact and improved productivity. So we aim to be a global leader in applied agri-tech, and to act as an independent nexus really between government, researchers and industry, and catalyse multidisciplinary and collaborative R&D to deliver on business needs and also to help the sector to grow. And we do that by hosting world-class open access facilities for collaborative R&D, and also by having different modes of engagement. So anyone can engage with us around grant funding opportunities and around commercial opportunities, or through our membership scheme. So just to very briefly overview the facilities or capabilities that we have these across the UK in partnership with key research institutes and universities and some businesses. And these really fall across our four themes. So our first theme that we work around in CHAP is soil health. And within that, we have a soil health and phenotyping facility at Cranfield, co-hosted with AgriEpi. And that really looks at the full rotation of agricultural production within a controlled environment in large lysimeter boxes with a simulated weather environment, and also with the phenotyping gantry, so we can do above and below ground phenotyping on these lysimeter boxes. As we grow crops in them, we can also drill or till in different ways directly into these boxes. So it really is a controlled mimicry of a field environment for soil health research. And we've also got field scale precision equipment up at Stockbridge Technology Centre. So we've got a strip till, and we're using that really to look at more sustainable forms of arable cropping, including leguminous cover crops within wheat and barley crop systems for crop health and protection benefits. And within our data diagnostic insights strand, we have a number of capabilities that are focused across crop protection and environmental protection. So we've got Crop Monitor Pro, which is our joint with Ferra. It's our pest prediction tool that was launched in September commercially, available to farmers, and that combines modeling data with predictive algorithms and with weather data, and also sensor data in a sensor network of fields to really predict crop health and disease threats. And we've got our eFlows mesocosm also at Ferra, which looks to understand dynamic risk assessment of crop protection products and within a more natural environmental system. So within crop protection, we've got a number of facilities, from our advanced glasshouse facility, which is specifically designed to look at biological and new novel IPM strategies, or integrated pest management strategies, within glasshouse production. And then right through to our fine phenotyping laboratory at Rothamsted. I think we've got Tom and our tutor from Rothamsted and CHAP joining us today, and they work and run our fine phenotyping lab at Rothamsted. And then controlled environment agriculture, we've got a number of exemplar facilities here from a semi-commercial ebb and flood vertical farm at Stockbridge, and a smaller collaborative research and development focused containerized facility up at the James Hutton Institute, the Innovation Hub for Controlled Environment Agriculture, Liberty Produce, and then down at Warwick in conjunction with Wright Building Services, we've got a natural light growing centre, which is an ETFE, advanced ETFE greenhouse, where we're looking at the effect of UV light on crop health, protection and production within controlled greenhouse environments. Just to really touch upon, I mean, we do a number of projects, as you can imagine, and just to really touch upon a couple of the projects that we've got ongoing at the moment that are relevant to phenotyping. We've got, and these basically are running out of our fine phenotyping lab at the moment. So we've got a blackgrass imaging project that is looking at multispectral approaches to differentiate blackgrass with wheat across different growth stages, and then potentially to integrate that capability into robotic systems. And we've also got our Slugbot project, which is Innovate UK funded project, and that's looking to develop a technology for autonomous monitoring and precision application of biomolluscicides for slug control. And there we're looking to differentiate slugs from background vegetation in order to really train a robotic applicator system there. And in terms of why we approached Phenome UK to have a collaborative workshop, it's really part of our forward-facing research and innovation program that we have set up at CHAP, which is called our New Innovations Program. And that really is a business case development program, a collaborative stakeholder engagement program that looks to intervene where barriers to sector growth are evident in particular areas, and develop an evidence-based and industry-led approach to prioritizing key challenges within that area and identifying solutions, co-identifying solutions, and then developing those solutions into a business case to lobby the sector, to lobby for public and private sector funding to tackle that market failure and help to drive sector growth. And this is really very much our future-facing element of what we do at CHAP. And we do it across a 7-stage process that we've designed that I won't really go into too much detail about. But essentially, we would like to consider pre-symptomatic disease detection as a topic, but we're aware that it's quite, it's very much an early stage topic. A lot of the really exciting research that's going on in this field is going on within universities right now. And we wanted to understand where the key developer challenges were, where the critical end user challenges were, and how collaborative partnerships could be developed around this area first, before we actually undertook a program, a stakeholder program on this. So this is the first stage really in this process for us, and we're really looking at understanding better how different emerging technologies from VOC detection, hyperspectral imaging, spectroscopic methods, and omics, tomography, and fusions of those techniques, how they are developing, how they can be applied to meet key end user challenges, and how we can better understand the barriers to progress within this sector. So that's really why we've come together to organise this workshop, and yeah, that's I guess pretty much me done in terms of an introduction. So I will stop sharing my slides now. And just really to say that what we're going to do now is head into our 1 hour plenary session. There is a chat function available to everyone so you can ask questions as and when they occur to you. And a couple of my colleagues will be monitoring that, and we'll put a few of those questions to the speakers during the plenary session. Also in the chat already, there's a link to a Jamboard that people can use, but probably now, sort of in the next break, to input information upon themselves and what they're doing in this area, so that we can collate a bit of an understanding of what's going on in this area. And I will be giving the speakers 15 minutes, and everyone, if I could ask everyone else to have their videos off for this, and then at 14 minutes I'll turn my video back on and I'll pop up onto the screen and give them a reminder that that they're running out of time, and that would be great. So yeah, what we tried to do today in terms of putting together the plenary session was to take some of CHAP's contacts in the phenotyping sphere and some of Phenome UK's academic contacts in the phenotyping sphere, and combine them to give what we hope is a good overview of current progress from and current perspectives from both sides of the fence. And what we're really trying to do here is encourage that level of understanding and to bridge that gap between academia and industry in taking this challenge forward. So the first, let me just stop my presentation on my own computer so that I can actually operate it. So the first speaker that we have is Keith Norman, and he's an independent consultant who has spent a long time prior to this working within agronomy at Velcor and similar, and so he has significant crop production and crop protection expertise, and is involved in a lot of industrial projects around phenotyping. So I've asked Keith to start with a bit of an overview from his industrial-based perspective. So I'll pass the baton on to you now, Keith.
Thank you very much. And if the technology works, hopefully you're now seeing my screen. So good morning, everybody, and thank you very much for the invitation to come and talk to you this morning. I've been involved in the industry for almost 40 years in terms of practical crop production and crop research through in-field trials and collaborative project work, looking at a whole host of different things. And I'm now an independent consultant and working with quite a few startups and companies as well, continue along that line of real crop applied technology and developing that. So I just wanted to share with you some of the aspects of new technologies that we've got in terms of pre-symptomatic disease detection. So just to start with, why is pre-symptomatic disease identification an advantage? And these are figures really literally hot off the press from AHDB, looking at septoria control, and these three fungicides that we have on this slide, Ascarilatis and Revistar, of, if you like, the Rolls-Royce fungicides. They are a mix of SDHI and DMI products. And you can just see that when you're looking at actual disease control, even with the best kind of combination product available to growers, you're still getting an advantage in terms of disease control when you're hitting something in a protectant capacity than if you're trying to cure a problem that's already established. So the basic principle is start early, you get a better effect. And there are also some nuances in as much that, you know, when you go in pre-symptomatically with a product, the actual choice of product becomes less critical, and some of the older, more commonly used products actually do work better in that scenario than when an infection has already started. So the first of these technologies I just want to look at with you is a project I'm working with Rothamsted and Blackheart, and this is a LAMP Assay DNA spore sensor. So just very, very briefly how it works. You can see there's an intake here where air is actually sucked into the trap. There is a process whereby the DNA containing in the spores of that air are smashed open and then put into solution with primers, and then that little file, that glass file with that liquid in, starts turning cloudy in response to how much DNA of the pathogen you've selected for is there. And then a light is shone through the file, and how much light gets through gives you an indication basically of the amount of DNA of the pathogen of interest. And you can just see this, the sort of printout you typically get. And you can see there's some, the most critical one here is the fluorescence line as the spore containing liquid is then warmed up and then the light is shone through. You can sort of see that the fluorescence is starting to pick up. But also to actually get an impression about how the disease might take hold, you need things like wind speed, temperature, relative humidity, rainfall. All those key weather metrics will tell you if the spores that you're detecting are actually going to start developing in the crop. So it's sort of a combined approach. This is a new project I'm working with the University of Manchester and SONY, which is a completely different concept, but very much going down the pre-symptomatic route again. Inside this housing here, where airborne spores just naturally flow through on the wind, there is an artificial leaf surface. And impregnated on this leaf surface are several biochemicals that are known to encourage the germination of a specific disease that you're looking for, whether that be yellow rust, brown rust, or in the vegetable world, downy mildew. And as the spore detects these biochemical stimulants, it starts to grow. And the hyphae that come out of the spore are then detected by this little micro camera that's looking down on this artificial leaf surface, and through AI that camera knows that those roots are from that disease because the way they grow, the time of the day they grow, whether they're growing at night or in the day, all of those little nuances are embedded in the AI of the product. This is another system from Photonics, which is a startup I'm working for. And the beauty of this is that you can actually put whole plant samples into this machine, which is pictured here on the right, or you can do single leaf layers themselves. So here we have RGB images of the leaf, and these are numbers of days after infection. And you can see that the Photonics system is starting to be able to pick out the severity of the disease quite nicely, whether it's on a single leaf layer or as a whole plant. And it just gives the whole plant image gives you a little bit more of an insight as to where in the plant the infection is. Is it on the new leaves, the older leaves? Is it spreading throughout the plant, or is it maintaining its position within a certain area of the plant? And these are some of the spectral data from that. And you can just see here that the different inoculation lines with the different times versus the control. And you can start seeing that the inoculated plants do behave at certain key wavelengths in a different fashion. And I just want to develop that for you because this is really where the hyperspectral technology comes into its own. This is some work that Hummingbird, again another company that I work with, is looking at pre-symptomatic disease detection. And this was looking at field trials here with a number of different varieties, with a number of different fungicides applied to them to generate, if you like, different backgrounds of infection pressure. And these are the various parameters that they're using. It's sort of NDVI, which is the norm really in the industry, but then there's red edge, there's green difference, vegetation index, et cetera, et cetera. And what they found is that, again, at key wavelengths, there are differences between plots that there are no disease and where disease has started but was not visible. And it's sort of these areas, these sort of 3 areas here for septoria, that they're now concentrating on as being perhaps the clue that something is going on within the plant that is giving a signal infection's taken place, and therefore enabling growers to get out there and start crop protection programs in good time. Another system is thermal imaging, and this was some work done at JIC. And you can see on the sort of the normal photo here of crop plots with different diseases, you can't really see an awful lot, but if you actually look from a thermal imaging point of view, you can see quite a bit of a difference between the different plots in response to the fungicide treatments they've had applied to them. And basically what happens is the more disease a plant gets, the canopy temperature, it starts to decline. And it's that decline that is the key to an infection starting. Now, there are lots of caveats here because you need to know a lot of, the best time of the day to do this, what the humidity is, because all of these factors can actually affect the measurements that you have. But basically, I've said here on the right-hand side, the sort of drop in temperature is anywhere between sort of 0.5 and 1.3 degrees compared to a healthy leaf. This is some work that was done on sugar beet, and it's really looking at 3 different types of, or 3 varieties of sugar beet in relation to its vulnerability to Cercospora. And again, a bit like the hummingbird and a bit like photonics, there are key wobbles in the spectral signatures at certain wavelengths for this disease. And you can just see here, the resistant line is the blue line, the red line is a tolerant, and the green is the susceptible. And so all of the different tolerances on a variety basis are starting to be teased out around that 1.8 to 2 nanometer kind of wavelength. And then there's another teasing out here at the 2.3, 2.4 mark. So again, key indicators of where to go and look for changes taking place that might indicate disease taking off of the disease you're interested in. This was quite an interesting piece of kit that was featured at the REAP conference. That I don't know if any of you were on that, but it's basically, it looks like a frisbee, quite cheap. You can put these in crop, and they're basically looking at the crop every hour of every day and making a huge range of different measurements, as you can see down here. It includes a 7-band spectrometer in the frisbee, but it's also measuring a lot of other things like solar radiation, humidity, soil moisture, wind speed direction, precipitation, dew detection, evapotranspiration. So all of that information can be modeled together to say, okay, we know that this specific disease needs these parameters to grow and develop within a crop. And so that piece of kit is basically on the alert for that combination of external factors and then early indications of a disturbance in the leaf itself from either the chlorophyll or through the spectrometer readings to say that something is going on. And these are all networked and go through a mobile phone app, so you can actually check in your fields every day what's going on. This is a new technology looking at volatile organic compound detection from the North Carolina State University. And it's basically a clip-on piece of kit to an iPhone, which is rather ingenious. And at the moment, it's very much down this stage of development where you would put your, and again, they've developed this for Phytophthora and tomatoes, but they're now developing it for late blight in potatoes. So you put your leaf in here, put a little tube in, it sucks the air in and gives you an instant analysis as to whether there are of the volatile organic compounds that are normally associated with Phytophthora present in your crop. And this is the latest technology, dogs, for pre-symptomatic crop disease detection. If you just look at the anatomy of a dog, they've got 300 million olfactory sensors in their nose. We've got about 6. And the part of their brain that analyzes smell is 40 times larger proportionally than ours. And there are 3 areas where, in fact, dogs are now being trained to sniff out disease. Clubroot in oilseed rape, by this fella. In the avocado industry, there's a disease called laurel wilt. And the big one here is in the citrus industry, particularly in Florida, there's a disease called citrus greening. And these dogs can evidently sniff out trees with citrus greening disease pre-symptomatically 2 years before symptoms appear. So that enables the orchard managers to go in there and grub those trees out, get rid of them, dig the roots up, and replant with a fresh stock before symptoms express, and then spread the disease on a much wider footing throughout the orchard. So other technologies which I haven't really got time to go into, there's obviously fluorescence spectroscopy. There is our ELISA-based lateral flow devices, and there's also work being done with qPCR, looking at qPCR detection, and then you will get a measurement of picograms of DNA for the disease that you're actually looking for. So really a lot going on there. We're very much at the early stages of an exciting digital revolution here in crop production in the UK, and a lot of these new technologies are all now coming into the frame, being validated and tested, and we as crop managers will be much more there'll be a lot more advantage now to us to make better decisions in terms of crop protection as a result of those technologies. So with that, I'll finish. Thank you.
Thanks very much, Keith. Very much to time, so thanks very much for that. There's one quick question which I'll just ask now, seeing as it's there. So, which is, what is the price point for an in-field sensor? So, how cheap does an in-field sensor need to be in order to be successfully adopted wide across the sector in the UK?
That's a good question, and it's a question we are asking in a lot of the collaborative research consortia that I'm working with. There's a bit of a conundrum because when technology is new, it's expensive, but it gets very cheap very quickly. We can see that with the iPhone. Evidently, if there were only 30 iPhones invented in the world, the price per iPhone would be something like £3 million. But because we've got so many, the technology is cheap and it comes down to several hundreds. So the same principle will apply in crop production technology as well. What I would say, and I'm saying to all the consortia, is that the price needs to be as low as possible to encourage more uptake and more use, because a single sensor is not an awful lot of good on its own, but a network of sensors is much more effective. And if you can, within any farming business, have 50 sensors throughout rather than just one, you're going to get much better data. And then there's also the possibilities of then connecting all of those networked sensors to other farms with their network sensors, and you start getting a much more effective background of good crop information that people can use. And the providers of these systems then have another opportunity for a sale, because they can sell the sensor, but they can also sell the service or the networking subscription. So really, my advice to everybody is keep the margin per sensor as low as possible to encourage adoption and uptake, to then get a much more effective network in place.
Thank you very much, Keith. So we'll try and limit it to one or so questions after each speaker, and then we'll hopefully have time for a couple more at the end. So I will move on to the next speaker now, which I see he's just turned his camera on, so he's definitely with us, which is Ben Curtis, and he's the operations director at Robo Scientific, and they are an SME that are developed in the VOC sensing and detection arena. So I'll hand over to you, Ben, now to give your talk.
Fantastic, thanks. I just need someone to allow me to share my screen, and then I'll be with you. So, morning everybody, anyway. Hope the weather's better for you than it is in Cambridge. It's absolutely miserable here today.
Claire, if you're there, are you able to provide a co-host for Ben so he can share his screen?
It's okay, we're in.
Wonderful.
Finally in. Fantastic. So I hope you can all see that now. And we should be off and running. Fantastic. Right, so I've seen a few familiar faces today, so apologies for those that have heard some of this before. But it was very interesting when Alex and Harry got in touch to talk about Phenome UK, and certainly the pre-symptomatic technologies going on, because obviously I think we all understand that early detection allows more rapid intervention, certainly more effective intervention. So I'll tell you a little bit about where we've sort of come from and what we do, and then I'll give you some examples of how we're commercializing that technology and using it in the field to aid the industry as a whole. We're about 4 years old now, and we originally span out the University of Leeds and then conducted an MBO, and we're now fully 100% privately owned, which is quite rare in the startup sphere. We use volatile organic compounds, or hereafter known as VOCs, to detect for disease, and VOCs very simply are smell particles, you know, that create odours in your nose, or the dogs, as Keith was talking about. Over the last few years, we've been developing with quite a lot of partners, both academically, both IUK with CHAP, and with some retail establishments as well that will be well known to most people. And well, this is 2018 data, and really the challenge is, you know, 750 billion per annum food wastage, and that's quite extraordinary. And we thought we had a technology that could help with this. We actually began working with humans. We still do a bit of clinical work, but we saw an opportunity in agriculture, and initially that was with poultry. And we went onto poultry farms and were able to find diseases in the chickens as we could in the humans. And then we started thinking, where else can this be utilized? And we looked at crops and growing crops. The wastage was a huge part of that, because obviously the earlier you can intervene or be made aware of a problem, and the more you can do to mitigate it. And so we turned our hand to crops. The way the technology works is probably a good starting point. Essentially, all organic bodies, if they're animals or plants, or they all respire, and they in various forms. And as part of this respiration, they emit odors or VOCs. Now, our software will measure these in down to parts per billion, probably about 10 parts per billion. And we will then take a digital fingerprint of that VOC output. Each disease we found has a unique fingerprint. So then the software can cross-reference the fingerprint reading we're getting with one stored in the database and tell you that you have botrytis or a mildew or a fusarium or anything like that. And this all happens in real time. It takes about 30 seconds to a minute to give you all the information you need to make a decision. This is what it looks like on the screen. This is just a water trace and a butanol trace. But as you can see, I think certainly with the orange line, which is the sensor 2, here you can see it changes quite dramatically. And we have 24 of these sensors running at any one time, and each one gives you 96 data points. So we are running quite a lot of information in a very short amount of time to get what we need out of it. The sampling process is absolutely key with VOC detection. The amounts of the VOCs are so small, especially in the air volume of, say, a glass house or a crop store, that that you really need to get that right. And we worked very hard to come up with a pretty bespoke and clever system, if I say so myself, that gives us very, very reliable and very fast and very effective results. We have found out that we generally, as an approximate rule, can detect a disease about 3 days before traditional microbiology, and certainly before visual symptoms or an agronomist becomes aware of them. We think we can get that more preemptive with dreadful grammar, but we think we can improve that significantly even more, and it's a very encouraging start nonetheless. The good thing is that now that we have all this information, we can use the VOC screening system, monitoring system, as a rule-in test for more detailed diagnosis. So whereas your PCR testing may take hours or days, we can tell you have a problem, then you can go back and by all means confirm that, but at least you know you can take action immediately. And sometimes that action is just as simple as we have 1,000 tons of potatoes that look like they're on the turn, let's just get them out the door now. And other times the treatment is slightly more involved, but it all aids to giving the farmer information. I'll take you through some practical examples and I'll just show you how it really works. This is a disease called Blackheart in Swede. We ran this in conjunction with ADHB. And Blackheart, by its sort of definition, you can't see it, it's an internal defect. And so the only way to really guarantee a positive diagnosis is to cut the Swede in half, which is pretty destructive. So we had a look at this and we found that we were able to differentiate infected, non-infected Swedes with 100% identification. This was repeated and has been repeated and is, a pretty effective demonstration of what we can do with this technology. We really are right on the cutting edge. Like all our sensors are made bespokely in-house, and they're quite significantly more advanced than the ones on the open market. And it's certainly, although we are new to crops, so probably a couple of years in, just starting commercialization at the moment, there really is a huge potential for this VOC technology to make a real difference. And we see that as collaborative with the industry. We see that working with other technologies and integrating with other systems as well. In terms of potatoes, here is a VOC comparison of healthy potatoes and potatoes with Blackheart. And again, you can see in the green ellipse, these are the healthy potatoes, and in the red are the infected potatoes. These graphs are a little bit counterintuitive because they're actually four-dimensional graphs represented in 2D. But all we need is a clear separation, so the software can then group those samples and see a clear difference. There's normally a step change when a disease happens, where you go from the healthy VOCs to a slightly more interesting mix, which gives you the diagnosis. Tomato plants. Now, tomato plants are fascinating. We did some of this work with STC, Stockbridge, and this is botrytis. And what you can see is that it's a much larger control circle than we got with the other crops. Now, this tends to be, there's quite a variability in the levels of tomato plants, but they do largely correlate as to clean and healthy. But as soon as the infection occurs, all of the VOC levels just dramatically cluster together and become incredibly dominant. And so you can see a really, really clear separation and a really step change when a disease such as botrytis takes hold. This actually seems to be the norm for crop disease. Certainly it's the same in mushrooms, certainly in strawberry and cucumber, lettuce, basil, and potatoes, to name just a few. And it's really quite fascinating that these, minute in terms of particulate levels have such a revelatory effect in terms of disease screening. So obviously all of this is very nice, but it has to go into something. And so we've got two products coming out. They were coming out this year, but I think we all know that this year has been a little bit challenging logistically. So they're now coming out in the beginning of next year. And one is the farm monitor. which is the poultry monitor, but the other one is the crop monitor, which looks exactly the same as this. It's just orange, and it's about the same size as a shoebox, really, not very big, and it works in a similar way to a smoke alarm in that it's constantly monitoring the environment for disease and airflow and humidity, temperature, all that stuff. And it won't really do anything if you're fine. If everything's hunky-dory, then you won't hear from it. It just churns away in the background doing its thing, and nobody really hears anything. As soon as it finds a disease, it will then immediately run those tests again. So that's probably takes about 2 minutes. And then if it is confident that there is a disease, it will then alert the farmer either to his phone or an email, or there's numerous ways via Bluetooth and on the phone network we can get to people, and it'll just say, yeah, we think you have a outbreak of Blackheart, or we think you have an outbreak of Fusarium in warehouse 6. Go and investigate. The beauty of that is then that as soon as the alarm is raised, you can start working your logistics and you can start planning your treatment should you desire. It's fully automated, so there's it really is about as easy as you can get, and it's pretty inexpensive. We haven't settled on a price yet, but we have an idea. We're talking thousands, not millions, and all the data is downloadable to your computer should you want to dig into it in any depth. I mean, we generate a lot of data every day, and so there is it's very useful to go back and see if there's a correlative effect between certain management events and then an outbreak of disease that coincides with that. So the device is either either just attached to the wall, or it can be overhung above the crops. This just enables us to stay away from any ventilation or fans or doors, especially. We want to get a good idea of the internal environment, and so we will run air through the instrument into a concentrator for anything from 5 minutes to an hour, just to make sure we're grabbing everything within the space, really. And it seems to work quite well. So far we've successfully completed some onion work with Fusarium, and tomatoes with botrytis and both powdery and downy mildew. Swedes we have brown heart, and basil we have mildew, as well as further potato and leafy greens trials, and ongoing at the moment, actually. So it's all looking very good from that point of view. The reason why we've decided to stay inside is, as I'm sure some of you will have realized and be ahead of me, is that outside presents a whole world of unique challenges, because if you've got particulates that are diluted into parts per billion inside a building, then obviously it becomes that much more difficult outside. We have been doing quite a lot of work on this and on various soils and things. We work very closely with Rothamsted, and we're working with Newcastle Uni on some other stuff as well at the moment, which is really exciting for us. But outside may not be as difficult as it was once considered, but it would still be a little way off. But it's certainly a very encouraging, very exciting time for this technology. And really, we're sort of getting towards the end now. The other products we have a laboratory instrument that's been on the market for a couple of years. It's been doing very well. And we have a couple of handheld VOC analyzers that will be coming out next year. The advantage of the handheld is it has all the automated processing of the crop monitor, but it's fully portable. You don't need scientific training for it. You can literally walk into a supermarket and grab a bag of onions and check that they're healthy. So it's there's quite a lot of potential for this technology, we think, moving forward. And with that, obviously, I'll leave my details up on screen for a minute. If anyone wants to jot them down, I'll take a screen cap, and I'll hand back to Harry. Any questions?
Thanks very much, Ben. again, pretty much to time, so that's great. I'll just ask one question that I can see from the chat, which is around whether there's any information on which VOCs are released at point of disease. Are they specific to diseases you're finding, or are they general plant responses to infections such as MESA? And
that's a good question. The way VOC sensors, you know, whichever family or you know type you use, they don't measure for specific VOCs. They measure for families of disease, so or families of VOCs rather. So you know, one will be for aldehydes, one will be for alcohols, and that way. The way that we do it, although we spent a lot of time initially in the first year or so, spending loads of money and loads of time on very complicated GCMS. And what we found is that it was very interesting, but it was counterintuitively, it was actually a bit of a hindrance to figuring out what was happening within the plant, because we were getting all this information and it was just a total overload. So the way, you know, now what you do is you look for certain key changes that you know are indicative of that disease, and you focus on those. And that's without telling you exactly how we do it, because it's complicated. Yeah, I think monitoring VOC concentration for pests, certainly that works. We've done that with various aphids and things, and it absolutely does provide correct data. It's quite a useful tool as well. Basically, anything that breathes can be detected with VOC analysis. Is the simple version.
Excellent. Thanks very much. In the interest of time, we'll move on to the next speaker, and that is Tim Bohr, who is a research consultant at ADAS. So, Tim, I'll hand over to you.
Thank you very much, Harry. I'll just try and share my screen. Yeah, can everyone see that? Okay, thank you very much. Yeah, thanks for inviting me to speak this morning. Yeah, I work for ADAS, and today I'm going to speak about a project or mostly speak about a project which we finished last year, which was developing sustainable management methods for club root. So this was an AHDB funded project, and which was led by SRUC with input from University of Sheffield, P3, LSPB, and Soil Essentials. So just a brief background to club root, for those who don't know. So club root is a soil-borne disease, and it infects brassica species. So it essentially works by when it detects a suitable host, it releases zoospores, which infect the plant roots and create characteristic ghoul-like symptoms, as shown in the picture, the small picture on the right-hand side. These ghouls affect the root structure of a plant, as shown in the background picture here. It can result in complete plant loss in severe cases, although more typically it tends to occur in small discrete patches. Yield loss from club root ranges from anywhere from complete yield loss to very little, but typically for every 10% of plants affected, it results in a 0.3 tons per hectare yield loss. So there are some challenges when it comes to club root, with climate change likely to favour warmer, wetter autumns, which is more conducive to the disease. It survives in the soil for 15 years, so it's very hard to escape brassicas in the rotation for that length of time to reduce the levels of inoculum. There's no chemical or biocontrol methods available, although there is varietal resistance, which is effective. And it is very difficult to know you've got a problem if you've only got a very small patch in a very large field, particularly the case in countries like Canada, where canola fields are very large. It can be very difficult to know if you've got a club root problem or not. So there are several aims of the project, but what I'm going to focus on is the bottom two aims, which is it was to establish through field mapping the impact and spread of patches of clip-root contamination in soils, and to investigate the cost effectiveness of treatments through field mapping. So of the fields looked at in this project for the pathotyping, 16 fields were selected for in-depth monitoring. So these were monitored in person. So each field was looked at 30 points per field, roughly on a grid pattern. And at each point, plants were assessed for clip-root disease. So they were physically pulled up and scored at each point. And through this, maps were generated from these fields to show where the clip-root was. And these fields were assessed at various times throughout the season. And through this, we found that clip-root patches were relatively stable. The blue dots are out on this example field where clip-root severity was low. The red dots are where clip-root severity was very, very high. This in-field monitoring matched very well with what could be seen from NDVI images. This is an example of an NDVI image from satellites, and but it is appreciated that the drawback is that a lot of things can influence NDVI other than clip-root. So you couldn't just use that as a way of diagnosing the disease. You still need to go out to the field and confirm that it is clip-root causing the reduced above ground biomass. From the in-field assessments, we could then generate economic treatment plans. So from every point we know the disease severity, we know the yield loss which would be caused by that disease severity, and we know the effectiveness of one of the few treatments that are available, which is liming, and the likely yield improvement from that treatment. And through doing that, you can identify which areas of the field it would be economical to treat and which areas would not. So in this example field, to the top of the field where all the green circles are, there would be a gross margin benefit from applying lime, but to a rest of the field, there would not be a gross margin improvement. It would be a negative effect. And we did this analysis on all the 16 fields where we've got intense monitoring data for. The column on the left-hand side, highlighted in red, shows the average margin over treatment cost per hectare for assuming the whole field was treated, and the column on the right-hand side in green is a margin per hectare assuming just the patches were treated, and at the bottom, highlighted in purple, is the average across all the fields. And as you can see, if all fields were treated with lime as a blanket application, there'd be a negative margin per hectare from the application. Whereas if the treatment was precisely applied, there'd be a positive margin benefit of around £30 per hectare. So the summary from this project was that clip-root patches remain stable throughout the season, and there is a potential for targeted treatment. But there is more research needed into the economics and rotational on-farm decision making, and much better detection of early disease is required. So this can be done without anyone going out to the field to validate if disease is there or not. And this brings me on to a new project which is starting in January, called Root Detect, which is looking at remote detection and precision management of root health. So this is funded by Innovate UK and the Canadian Research Council. It's being led by Airborne Robotics and SII Canada and ADAS. So this slide just summarises the importance of canola to Canada and to Europe. So the crop brings in around 15 billion to the Canadian economy and about £8 billion to the European economy. And the project recognises that clipper root is an increasing problem worldwide, and it's very difficult to manage once it is present. And early and rapid detection is essential for robust management. And if we could precisely map the affected field patches, it will enable targeted and economic treatment to be applied. So the project objectives are to develop a detection tool which is able to scout large areas quickly and cost effectively, to detect the very early emerging clipper root patches, ideally before they're visible from the naked eye, and to map these patches, and also to develop an early warning system to alert plant breeders when a cultivar resistance is potentially failing. The second objective is to link the detection and decision support systems with precision farming technology so that targeted treatment is possible. And third is to drive technical awareness through engagement. And yeah, this is being done by a series of work packages. So airborne robotics are taking the lead on the hardware side of things. So we'll be developing a bespoke autonomous or a semi-autonomous drone system to collect the high-resolution data. ADAS will be collecting the ground truthing data and assisting with the analysis and zone mapping and predictive analysis. SI Canada are taking the lead on the software aspects of a project and the novel aspects of a project, which is generating the algorithms which can determine the spectral signature of clipper root to enable the automated recognition of a disease. Yeah, and finally, the final work package, develop a user-friendly interface for the root detect system. So yeah, that's everything from me. Thank you very much.
Thanks very much, Tim. I've got time for one quick question, and that's from Alex, one of my colleagues at CHAP. So does disease severity alter the amount of lime that you would apply at a specific patch, or are you applying a standard rate?
In the research which has been done in the past, it is typically applied at a standard rate. I think that a lot of the cost of lime is involved in the transport of getting it to the field and in the application. So I think it is most likely that a robust rate would be put on when the field is treated.
Thanks very much. And I guess a slight follow-up from me. Was there any sort of topographical signatures? It was difficult to see from that, from those images, whether there's any sort of topographical signatures that were potentially affecting the prevalence of the clipper root in particular areas, like sort of low points in the land or whatever.
Yeah, it was very difficult to. It would be, yeah, in the project when we mapped the fields in person, the areas where the clipper root actually was different to probably where you might expect. So you tend to expect you'd probably find a clipper root in the gateways where soil's been transferred on farm machinery and the like, but actually quite often it was nowhere near the gateways and things like that. So yeah, it was very. So no, we've not found anything specific at this point, no.
That's great, thanks very much. Excellent. So it looks like, well, we're about 11 o'clock now. So in the interests of time, what I will say is thank you to all of the speakers for some really fascinating and really well spread talks across different phenotyping technologies that are perhaps more applied to earlier stage and with different focuses that's really set the scene for hopefully some really productive discussions.